Today, I will provide an overview of an ongoing collaborative project that our team is undertaking. Suzanne Needs-Howarth, Alicia, Hawkins, and myself are all zooarchaeologists. Louis Lesage is a biologist and a member of the Huron-Wendat Nation. Louis is also a hunter and a member of the National Aboriginal Council on Species at Risk, and thus brings a diverse perspective to the project. Guiry is a specialist in isotopic analysis of archaeological fauna, while Tom Royle is a specialist in the analysis of ancient DNA from such remains. Finally, I note that we have previously presented much of this data at the International Council for Archaeozoology's Bird Working Group meeting held online this past June. We would like to acknowledge that the land on which this research was conducted encompasses the traditional territories and contemporary homelands of many Indigenous peoples whose presence has shaped the history of this land for thousands of years. I'm joining you today from the University of Toronto, Mississauga, and wish to acknowledge this land on which the university operates. For thousands of years, it has been the traditional land of the Huron-Wendat, the Seneca, and the Mississaugas of the Credit. Today, this meeting place is still the home to many Indigenous people from across Turtle Island, and I'm grateful to have the opportunity to work on this land. The passenger pigeon was once the most plentiful bird in the eastern woodlands of North America, representing an estimated 1 in 4 of all birds, according to 19th century observers. Its population may have exceeded that of every other bird on the planet. While our understanding of passenger pigeon diet is incomplete, they largely specialized in the consumption of tree mast, primarily beech nuts and acorns, which are cyclically abundant. The species declined rapidly through the late 19th century and became extinct in 1914, when this last individual, a female named Martha, died in the Cincinnati Zoo. Two particular sources have influenced our interest in and approach to the current project. Our interest in passenger pigeons was inspired in part by this 2013 book by avocational ornithologist Joel Greenberg, who really captures the sheer awfulness of the passenger pigeon's extinction. The topic of our paper was inspired in part by this 1985 article by Thomas Neumann, which argues that the passenger pigeon was much more numerous in the colonial era than it was in the pre-colonial era, because the system destabilization brought on by European-style agriculture was actually favorable for the passenger pigeon. We aim to investigate this proposition in southern Ontario, using a meta-analysis of archaeological faunal data from pre-colonial contexts. The current meta-analysis is very much an ongoing project, which we have just begun to pull together over the past year. To date, we have compiled data from our own analyses, from published sources, and from selected reports by zooarchaeological consultants. We have also included 87 reports by undergraduate students in Dr. Howard Savage's faunal class at the University of Toronto, which are summarized in this book. Those of you who are aware of our work on fish know that we've been hesitant to work with learner-generated data, a topic that Alicia will address in the next paper in this session. However, with passenger pigeon, we feel a bit more secure, because passenger pigeons are osteologically quite distinctive compared with the only other locally common bird of the same size, namely grouse. To improve our chances of comparing apples with apples, we have excluded surface-collected assemblages. Our initial meta-analysis includes 171 Iroquoian sites in southern Ontario. Note that the distribution and density of the sites shown here is in part a function of research intensity, which usually equates to intensity of urban and infrastructure development, especially on the North Shore of Lake Ontario. These sites span the 10th to the mid-17th century CE, color-coded here by century bin, and with few exceptions are single component sites likely occupied for no more than 30 years. We have additional data for earlier and later time periods and for ubiquity, as well as data from sites in New York State and Quebec, which we plan to use in the future. This slide shows the number of bird bones identified below class, color-coded for whether passenger pigeon are present, the pink circles, or absent, the purple circles. Collectively, these sites have over 14,000 bird specimens identified below class, of which more than 8,000, or roughly 57%, are passenger pigeon bones. For reference, the small map at the top right shows the accepted primary breeding range in green and the primary foraging range in blue, recorded in historic sources. The breeding distribution largely corresponds to the limit of the mast-bearing forests, as you can see from the inset map. The old map at the bottom right shows nesting locations documented by Mitchell in 1935 from records and interviews related to the late 19th century. We hope to eventually plot the Mitchell data in GIS to explore it in more detail. Today, we present some preliminary trends that have arisen from our initial considerations of these data. We will also touch on aspects of future research that we anticipate exploring. You can see here that while bird remains are ubiquitous in our study area, bird, shown in the sort of maroon color, is consistently a very small proportion of faunal assemblages compared with fish in blue and/or mammals in pale orange. Regardless, birds are abundant enough to consider patterns in the relative abundance of various bird taxa. Note that this is a somewhat reduced data set shown here. We have data on all classes for all sites, but because of lockdown restrictions, we haven't yet been able to access the student reports to input the non-bird abundance data. We've been exploring passenger pigeon as a proportion of total birds identified below class across both space and time. We've also been considering proportions of other migratory birds, such as geese and ducks, and upland game birds that do not migrate, such as ruffed grouse and wild turkey. We feel that ducks and grouse, being comparable in size to passenger pigeons, are particularly useful comparators because they would have likely been subject to similar taphonomic processes. In contrast, turkey and goose skeletal elements were very commonly used as raw material for the manufacture of tools and items of adornment, and thus their representation in these assemblages is more variable and less comparable. Here we show the relative abundance of birds across our study area. While grouse in red and ducks in blue are variable on the site level, they're relatively consistent on a more regional level, with the exception of those sites in the very different environment along the St. Lawrence River. This consistency reflects the nature of these taxa as more consistently available, locally constrained resources, with people able to predictably hunt these year after year in numbers that local environments could produce. On the other hand, passenger pigeon in green are more spatially variable, but this is averaged out in this plot over the long times represented by our data. People would have responded to this unpredictable but hyperabundant resource by focusing hunting effort on them whenever they became available, possibly from short-term hunting sites. One possible way to explore whether people were mobilizing their hunting effort in response to passenger pigeon resources is to consider whether the archaeological record contains evidence for passenger pigeon hunting camps. As a preliminary perspective on this map shows sites that have been identified as villages in light green versus non-villages in dark green. Importantly, this reflects the site designations assigned by the excavators. We have not yet applied a standard definition of villages versus other site types. The size of the circles here reflects the numbers of passenger pigeon elements identified. While there are some non-village sites in our data set that have no passenger pigeon bones, a few, particularly around the western end of Lake Ontario, have higher numbers. Here we show the relative abundance of passenger pigeons compared to grouse and ducks in these non-village sites, with the size of the circle reflecting the total sample of identified birds. Notably, the two sites with the largest samples of identified birds are both heavily dominated by passenger pigeon. These may then be good candidates as specialized passenger pigeon hunting camps, though we have not yet been able to access fish or mammal data for these two sites, and thus cannot rule out that other hunting or fishing activities were also important at these locations. Here we map bird abundance over time, with our sites binned by century, and with the smaller sample of pre-14th century sites lumped together. The dates for these sites are based on ceramic seriation and/or radiocarbon dating. While there are certainly many aspects of these data for us to consider, we've been particularly struck by the clear decline in passenger pigeon abundance, in green, in the 15th century. We cannot yet explain this pattern, but there are several possibilities that we plan to explore further in the future. Variable human population could provide an influence here in terms of the hunting effort that could be mustered in response to the appearance of this hyperabundant resource. As a coarse initial perspective on this, we note that the relatively low numbers of passenger pigeon in the 15th century correspond to the end of the 14th through early 15th century Iroquoian population boom in the area discussed by Warwick. We plan to examine demographic trends on a finer temporal scale in the future. For the 16th and early 17th centuries, for example, demographic information and the temporal resolution of the archaeological data can be more finely constrained because, in part, of the sub-century dating potential of glass trade beads. These plots show mean passenger pigeon abundance as a proportion of total identified bird on the left-hand graph, and as a portion of total identified duck, grouse, and passenger pigeon on the right-hand graph. Error bars show the standard deviation. While passenger pigeon abundance is variable, these plots again clearly show a decline in mean passenger pigeon abundance in the 15th century. In addition to possible demographic factors, this time period spans the transition from the earlier medieval climate anomaly, a period of relatively warmer temperatures, to the later Little Ice Age, thought to start around 1450 CE locally. It is possible that these climatic changes influenced the local production of tree mast. This may not have affected passenger pigeon populations on a continental scale, but may have limited the northward distribution of passenger pigeons in the breeding season. We plan to examine these factors more in the future. One complicating factor is that our previous stable isotope analysis on bone collagen indicates the consumption of maize by passenger pigeons. The graph at the bottom left here shows Iroquoian sites in yellow and settler sites in red. As illustrated here, carbon and nitrogen isotope values produce a tight clustering for the majority of passenger pigeon samples, consistent with a diet comprised primarily of tree mast. Importantly, a second cluster of 6 individuals from Iroquoian sites at the top right of the graph were clearly consuming significant quantities of C4 plants, almost certainly maize. Because bone collagen remodels very slowly over the life of an individual animal, these data speak to lifetime average dietary trends. So some individuals were clearly specializing in maize. Unlike tree mast, maize may have been a predictable and locally abundant food source for passenger pigeons, which coincidentally also makes them easier to hunt, as they feed and roost close to villages. Think of it as pest management with benefits. People would be able to predict this. In years of low mass production, watch out for pigeons in your fields. Our previous work also involved some small-scale ancient DNA analysis. The temporal network shown here compares our data from 13th and 14th century sites with data from 19th century museum specimens. The 2 most common identified haplotypes are shared between the earlier and later samples. One 14th century specimen has a unique intermediate haplotype not observed in the 19th century. This may reflect greater genetic diversity prior to European settlement, though calculated haplotype and nucleotide diversities for these populations are similar, suggesting they have comparable genetic diversity. We may seek to expand our isotopic and aDNA sampling in the future to further explore these patterns. In addition to the future lines of research I've already mentioned, we would highlight a few additional plans. At this point, our data is relatively coarse. We aim to explore more fine-grained temporal bins for example, using median dates, and provide more fine-grained analyses of the faunal data. We've also discussed whether we can determine if a site was in the vicinity of a nesting colony. Specifically, can we identify bones of unfledged squabs, such as that pictured here, as opposed to those of other juvenile individuals that were able to fly? We also hope to provide more nuanced considerations of spatial patterning, such as examining the distance from major water bodies and considering possible locations of major fly routes. Finally, we hope to widen the temporal and geographic scope of our data. For example, there are 19th century nesting records from the Canadian Shield, further north from our study area. The problem here is that we have very few faunal samples from those areas, as excavation coverage is largely determined by development. And bones often don't preserve well on sites on the Shield. Broadly, we aim to holistically explore the intersection of environment, bird biology, and human social organization as they relate to passenger pigeon abundance and use in the past. To conclude, we return to Thomas Neumann's suggestion that the passenger pigeon was much more numerous in the colonial era. The difficulty with all faunal analysis is translating bones into biomass. We have some sites with hundreds of passenger pigeon bones, and compared with other birds, this does indicate at least local abundance. We have shown that passenger pigeons were fairly ubiquitous, but that they show quite a bit of variability in time and space within a 700-year span in the Ontario portion of the core breeding area. For now, we have not directly examined colonial era data for our region. We haven't settled on explanations for the observed variability, but plan to continue exploring this as our project progresses. This project provides a unique opportunity to use the archaeological record of an extinct bird species as a dynamic link between other scientific disciplines, like historical biology, ecology, and the anthropology of Indigenous peoples. All of this can help us to better understand the not-so-distant past and the life and times of Indigenous ancestors. Thank you for listening, and I'd like to thank these individuals and institutions that have helped support this research. Thanks.